Hello everyone. Today I am going to discuss the fundamentals of Avarana. Avarana is a specific terminology used in Roga Nidana in different aspects. So literally the word Avarana means a covering or obstruction. When you look into the contextual meaning may have some different aspect. For example, ignorance also may be there. But medically we can say there may be a possibility of obstruction or covering. So before going into the description of Avarana, let us know something about the concepts of Avarana. So what are those concepts? Basically three concepts we need to have to keep in our mind. First one, if the physics, chemistry and biology, fundamentals of Ayurveda are well known, then every information in Ayurveda as well as Roganidana can be learned with deepest understanding. Second, we have to practice the concept of minimalism. What is the word minimalism? Minimalism in the sense one particular word, think from multiple angles. In other words, we are doing the brainstorming of that particular word. So third, if you learn the concept of Kriya Sharira, you can learn the concepts of Roganidana well. If you are knowing the concepts of Roganidana with much deeper understanding, we can have we can learn the concept of Kaya Chikitsa even with much deeper understanding. With this particular concepts in our mind, so let us look into the concept of Avarana. So when you look into the physics point of view, so here I will be giving you an example. So here a table is there. In that particular table, three objects were being kept, P, Q and R. P is having the very small weight, Q is having the moderate weight, R is having a larger weight. You are being asked to toss these objects into the sky. So which one that will be moving to the sky to the maximum level? Obviously P. R will be moving very less height and Q will be moving in the between the P and R, right? So same way, when you look into the other example, you have football, you have cricket ball. Football, if you toss the football, that is not moving much higher. Whereas if you are tossing the cricket ball, that will be moving very much taller or heighter than the height of the distance of the arc. So the same thing can be applied even into the blood vessels also. So when you look into the green color of this circular structure, if it is, let it be a blood vessel, it is actually the origination of this pressure that will be from left ventricle. So left ventricle pumps the blood, which is having different components of various molecular size. So sodium, a small molecular size. So WBC is somewhat a moderate one. Plasma protein somewhat a higher molecular size, macromolecular size. So that means, so in the blood vessel, the electrolyte that will be moving very fast. So WBC that will be moving a little bit slower. Plasma protein that will be moving very slow. And the higher molecules, that is the larger molecule, they usually travel along the wall of the vessel. That is, whereas the lighter molecule that will be moving in the center of the vessel. So when you think about the blood vessels, they are having three categories: arteries, veins and capillaries. Arteries, they are carrying the blood from the heart to the different parts of the body. So for example, aorta and pulmonary artery. Whereas the veins that will be carrying the blood towards the heart. For example, the inferior vena cava, superior vena cava and pulmonary veins. And the third category of the blood vessel, that is capillary, which is considered to be the major portion where the majority of the functions of the cardiovascular system that will be taking place. That is nothing but the transport of nutrients, metabolites, gases, that is the oxygen and carbon dioxide across the capillary network. Here there is a golden nugget that we need to learn, we need to remember, that is human body, it is composed of about 10 billion capillaries. 10 billion capillaries. So that means there is no other space that will be devoid of capillaries. For each and every minutest structure, there will be the capillary that will be seen very near to that. So, what is the function of capillary? Well, before knowing into the functions of the capillary, we need to know about the functions of the Starling forces. What is the Starling force? Starling force, they are nothing but the pressure gradient that is being observed. Let us say this yellow colored structure, let it be the capillary, and the blue colored structure, let it be interstitial space. So, and green colored arrow, that is depicting the hydrostatic pressure, orange colored arrow, that is depicting the oncotic pressure. Now we are going into these different pressures little bit in detail. Whenever if you are saying Starling forces, basically two types of forces that will be acting across the capillary membrane. So one that is being generated by the water portion, second one that will be generated by the proteins and electrolytes. So the pressure gradient or the pressure that is generated by the water portion, that is being termed as hydrostatic pressure, and the pressure that is being exerted by the proteins and electrolytes, that is being termed as oncotic pressure, and tissue tension is nothing but the hydrostatic pressure of the interstitial space, and oncotic pressure is also being present in the interstitial space, so which will be actually generated by the presence of proteins and electrolytes in the interstitial space. So whenever there is the blood pressure, if it is 120 by 80 millimeter of mercury, this will be the pressures, hydrostatic pressure as well as oncotic pressure that will be observed in the capillary as well as interstitial space. What is the function of the case? Different pressures. Hydrostatic pressure is actually responsible for the movement of the fluid from capillary to interstitial space, whereas oncotic pressure of the capillary, that is responsible for the pulling function, that is reversal of the functions of hydrostatic pressure that will be taking place. Whereas the hydrostatic pressure, that is usually being termed as tissue tension in the interstitial space, that is responsible for the push function, that is movement of the fluid that take place from interstitial space to the capillary, whereas oncotic pressure of the interstitial space, that is actually retaining the fluid portion in the interstitial space itself. When you look into the overall the functions of capillary interstitial space, these hydrostatic pressure, oncotic pressure, tissue tension and oncotic pressure of the interstitial space will not be functioning one after the other. They will be functioning all together at a particular time. For example, whenever if you are looking into the arterial end and its effect of the Starling force at the arterial end, so that is both of these hydrostatic pressure as well as oncotic pressure, they act together, whereas in the interstitial space the tissue tension and oncotic pressure of the interstitial space they act together. When you want to calculate the net pressure gradient, we have to summate these two different types of pressures observed in the capillary as well as interstitial space, which is being mentioned in this particular image, that is the hydrostatic pressure and oncotic pressure of the capillary minus hydrostatic pressure and oncotic pressure of the interstitial space, which amounts to be 11 millimeter of mercury. That means with this amount of pressure, whatever the nutrients, water portion that will be moving from capillary to interstitial space. Now, let us come to the description of the venous end and interstitial space. When you look into that, so again the formula is the same: hydrostatic pressure and oncotic pressure of the venous end of the capillary minus hydrostatic tissue tension plus oncotic pressure of the interstitial space. So that amounts to be minus 9 millimeter of mercury. So that means with that amount of pressure, the water portion metabolites of carbon dioxide, so that will be moving from interstitial space to the capillary. So now, if we have learned about this, what is the purpose of knowing this particular aspect, concept? Whenever if you look into this concept of Starling forces, whenever if you see the arterial end of the capillary, there will be more amount of the fluid. Whereas at the venous end of the capillary, there will be less amount of fluid. So that means, so when you compare the arterial end of the capillary, venous end of the capillary, the venous end of the capillary will be showing lesser movement or slower movement when compared to the arterial end of the capillary, because the water portion is being observed in the arterial end of the capillary, that is much more, whereas in the venous end of the capillary that will be lesser. That is the reason. And why this particular information is necessary? Because when you say this particular movement of the fluid etc. in the venous end of the capillary, that is being termed as sludged blood. Sludged blood in the sense what? So when you think about the arterial end of the capillary, if it is similar to the consistency of the milk, whereas venous end of the capillary probably it could be the consistency of a honey. So that is when you think of these, if you are taking a tubular structure, which one will be moving easier along the tubular structure, milk or honey? Obviously milk, that will be moving faster than the honey. So whenever if the because of the one or the other reason, if there is any pathogens that will be present in the interstitial space, the movement of the fluid etc., WBC etc., that will be moving more into the interstitial space from the capillary. So that is the reason whenever if there is any infection or inflammation in the interstitial space, that will be responsible for the thickness of the blood in the venous end of the capillary, which is termed as sludged blood, and this is the initiating factor for the manifestation of avarana. Now, regarding the applied aspect of sludged blood, what is this particular applied aspect of sludged blood? So that means these are the different mechanisms wherein the edema will be manifested. So edema will be manifested either in the serous cavity or either in the interstitial space or either inside the cell, right? So there are basically 5 different mechanisms by which the edema can happen. One, that is increased hydrostatic pressure of the capillary. So that means if the fluid portion is more in the capillary, or otherwise if the blood pressure of the left ventricle is much higher for 170 or 180 millimeter of mercury, in that particular situation we may get the edema manifestation. Second, decreased oncotic pressure of the capillary. Because of one or other reason, if there is a decrease in the proteins, maybe liver disease or maybe protein malnutrition etc., or if there is any reduced electrolyte imbalance, that is reduced sodium ions, definitely that will be responsible for the again edema manifestation. Third category, that is capillary membrane damage, maybe because of trauma or maybe because of some kind of the infectious exposure, and like that in that particular situation there will be total damage that will be taking place. So in this particular occasion, even the RBC sets that will be moving into the interstitial space, resulting in the reddish colored swelling, which is considered to be inflammatory swelling. Fourth one, that is lymphatic obstruction, because of one or other reason. For example, in case of filariasis or otherwise in case of lymph node resection, in those situations the person may have lymphatic obstruction. This may also be resulting in the edema or swelling. And the last category, that is retention of water and sodium in the body because of one or other reason, maybe because of dehydration or maybe because of the renal diseases, etc. In those situations, there will be possibility of the edema manifestation. Suppose if this is in the schematic representation of human body, if there is any sludged blood is happening, in the right side we have seen it is a normal structure schematic representation of human body, wherein this will be a gastrointestinal compartment, intravascular compartment, interstitial compartment, and intracellular compartment. Whereas in case of abnormal Starling forces, this intravascular compartment may become narrow, which may be resulting in the swelling of the either interstitial space or intracellular space or alimentary canal. So this may be resulting in a specific clinical manifestations observed at a particular area. For example, if the person is having the exposure to dust, what will happen? So that is responsible for the increased blood circulation towards the area of the lungs or bronchi, where there will be more and more secretions taking place into the interstitial space, resulting in a narrowing of intravascular compartment at that particular region, which results in the widening of interstitial space of or interstitial space of the bronchi or lungs. This is what is considered to be will be happening in cases of the abnormal sludged blood. This can be generalized to every other aspects of a part of the body. One take-home message is Avarana is a pathological phenomena. Whenever there will be the presence of the sludged blood, so that will be an initiating factor for the Avarana manifestation. This can happen in the intracellular compartment, interstitial compartment, intravascular compartment, or even this can be seen in the gastrointestinal compartment also. This may happen at the cellular level as well as capillary level, and it is usually unnoticeable initially in cases of cellular as well as capillary levels. So this is just a beginning. This is not a complete knowledge. This is the description of Avarana mentioned here is just like a drop of water from the ocean. Still, a lot has to be discussed in the context of Avarana. So, with this, I am stopping this particular video. Dear members, what were the major key takeaways from this particular video, and tell me about the one major area where you would like to have a discussion through video mode, and share this particular video to maximum number of people who want to learn concept of Avarana. Thank you.